Okay, so good afternoon and welcome to the colloquium. It's indeed a great pleasure to have Dr. John Katsaras here today to deliver a colloquium. He was here, I think, 12 years ago, and we hope we don't have to wait that long for his next visit to Bangalore. John obtained his PhD in biophysics from the University of Gulf in Canada, and then he spent a few years in France doing postdoctoral work. Then he came back to Chalk River and joined there in the neutron scattering facility there as a principal scientist. And as you will hear from him later, this is a facility which has played a big role in the development of neutron scattering, especially the early days, both the techniques and the understanding of inelastic scattering from solids. So John and his group there spent did a lot of detailed experiments on many things, many soft materials, especially on what are known as lipid aggregates called bicelles, which are widely used as substrates for membrane proteins and things, NMR studies of membrane proteins and things like that. After 2010, he moved to Oak Ridge, where now he's a distinguished R&D faculty. His work at Oak Ridge initially days were again related to neutron scattering, and they did some very exceptional work on understanding the origin of microdomains in lipid membranes. Now, recent years he sort of moved away from neutron scattering, and his interests are in using lipid membranes as neuromorphic platforms to understand biological learning and memory. I think today he may not have time to talk about the last bit. I think he's going to talk mainly about application of neutrons in biology.
Well, thank you very much, Raghu. I appreciate that. Well, thank you very much for taking the time to come to this seminar. Yeah, I did talk about these neuromorphic materials, Ganapathy's workshop just a little while ago. And I'm open to discussing some of these things after the seminar. But hopefully, we'll get things started here and then, take things for, after we're done. So I'm going to concentrate on neutrons. I thought I was going to extend the talk to be a little more than that, but then I rush, and then I don't get anything done. So I'd rather stick with this, and then we could take things in different directions. So I'll start off with the person that actually discovered the neutron, which was James Chadwick in 1932. So, I'm sure not everybody knows when the neutron was discovered. But James Chadwick used to be a student of Rutherford's. He was in Rutherford's lab when this discovery was made. And the experiment that he did was not dissimilar from many of the other experiments that were done by the Germans and the French at the time. In fact, I think the polonium came from France that he used. So what was the difference between Chadwick and all the others is that in the late 20s, Rutherford had actually postulated the possible existence of a thing called the neutron, and I think he even termed it the neutron. So, in other words, Chadwick was sort of aware of something like that existed, while everybody else only knew of the existence of gamma rays, electrons, protons, etc. They certainly didn't know about that. So whenever they saw an effect that he saw, they always attributed it to being something like gammas, because that's all they knew. So the interesting thing is that the setup was very similar. They used alpha particles from polonium. Then they interact with beryllium, the unknown radiation, which ended up being neutrons. And then they were detected by an electronic counter, which we now know as the Geiger-Müller counter. And in fact, Geiger himself was at the Rutherford's lab at that time. So they were all there. Rutherford was certainly one of the most influential scientists in the 20th century. Certainly, as far as influencing people, he was the most influential scientist of the 20th century. And promoting people, he was a great promoter. In fact, I was just telling with Raghu, he was a great promoter of Raman when Raman was trying to establish himself. So the neutron then was discovered, and the paper actually was very short. As I say to people, if you have something very important to say, it doesn't require much space to write it in. People understand it very quickly. And he was given the Nobel Prize very quickly in 1935. And in those days, of course, people didn't share the Nobel Prize as they do today. You know, before, everybody used to get it as a single prize. I don't know when that trend changed, but at some point it did change. And now it's usually very unusual that somebody, less than 2 people, will get the Nobel Prize. Can't recall of any, at least in science, that is given to a single person. So things moved very fast because people, you know, this became a very hot field, and people like Enrico Fermi had already were doing things that were related, and of course many things were happening in the 40s, and there was upheaval in the world, in Europe in particular, of course due to the Second World War, and many scientists from Europe ended up migrating either to Western Europe, to Britain, and in many cases to North America, to the United States and Canada. In fact, the Chalk River Labs were part of the Manhattan Project with Canada running the heavy water experiments of the fission experiments. So it was very interesting then that the field was progressing at a good clip, and we have then the first nuclear reaction at the University of Chicago in 1942. So it's interesting, this thing was constructed using fuel and absorbers in a squash court under a football field in the middle of Chicago. So that's where it was. There are no pictures actually of that. So there's no actual photograph. Everything that we know of this are models. These are the people that were part of it, at least some of them. Fermi is over here. And these things, and reactors back then, they were not called reactors, but they were called piles because effectively you used to just pile fuel, and then you would have some cadmium rods strategically placed in case things went, like they would say, went south. So there was even a person supposedly with an axe and a rope, and if things went bad, then Fermi would tell him, drop that cadmium rod in there, like ASAP. Supposedly before once they were set up, they went for lunch, and then they came back and they did things. So they were not in a rush, but he was Italian also, so you have to have lunch before you do anything important. So things happened then very quickly. We have Chadwick here in 1932. And then there are, these sources, which are basically hunks of rock that are now emitting neutrons. And then we have the first sort of machines that are producing them. Meaning we have the cyclotron, we have CP1, which is Chicago Pile 1, Chicago Pile 2, etc. And then we start having, in Canada, X10. So X10 becomes then the most powerful reactor, and where? In Canada, not in the US. And then NRX is the next big step. No, X10 X10 is in Oak Ridge, actually. So if you're taping this, I'm in trouble. If the Oak Ridge people see it, then I'm done for. X10 is the most powerful reactor of its time, and it's in Oak Ridge, and it actually exists today. I mean, it's not functional, but you could actually see it. And then in Canada, you get NRX, NRU, which I did a lot of my work, and then the reactors in the States, like Haifa, and then in France, the ILL. But it's interesting to follow that when the technology matures, then where do you go? As you could see, the flux here is basically plateauing. There's a factor of 2 maybe, but that's not a lot when you, in the scheme of things. And then what you have are the machines, then the accelerators, where you're producing now neutrons using accelerator sources. And this is something that we have at Oak Ridge. The spallation neutron source is an accelerator source. The ISIS facility, I'm sure some of you may have visited the ISIS facility in the UK. That's another accelerator source, and so on and so forth. But so we're moving in accelerator source for various reasons, getting away from reactor sources. Part of it is political. As you can imagine, it's hard to convince to build a reactor because of concerns that people have with reactors. So things then developed, and in the US, there was the elastic scattering part of neutrons that was being developed. They were at the forefront. And effectively, there was Ernie Wallen, who was one of the first people actually to start doing that at the X-10 reactor. And this is a proposal from Wallen to the then director of the science division, asking for beam time and facilities and help. So it's not a very long proposal. Today we'd have to write 30 pages to do the same thing that they did in one paragraph. And then we have, in Canada, at the Chalk River Labs, then you have Brockhouse developing the inelastic part using the very well-known machine now called the triple axis. And probably the most common machine found in a lot that physicists use it's a very common machine. Now maybe they're getting, the hard condensed matter people are getting away from the triple axis, but it's still a staple of scattering, is the triple axis machine. And it's a great way of doing inelastic scattering. So they won the Nobel Prize in 1994. That was the year I went to Chalk River. And I remember Brockhouse coming to Chalk River to celebrate the Nobel Prize. And it's the only, as far as I know, it's the only neutron scattering Nobel Prize given. That's the only one that I know of. And Wallen did not share in it because he was dead, so it was only Scholl and Brockhouse that got the Nobel Prize. So what are neutrons? So here we have our kettle that's going to be brewing some coffee for us. And as I'm going along, you'll see it sort of bubble. And some of you may pay more attention to that than to what I'm going to say. So what are neutrons? Well, they are neutral particles, and as such, they don't interact extremely well with a lot of materials. And that is good and bad, because it's good, in other words, that they can penetrate, but of course, then the signal is weak. So you have to balance then the fact that they can actually penetrate through a lot of materials, but then the signal is weak. They are non-destructive. So you don't have to cut something up. As you can see here, we're able to see the mechanisms of brewing coffee in this kettle without having to sort of do a lot of fancy stuff like cutting it apart or putting little windows. You could actually see everything quite well. And they are, because they have a magnetic moment, they're very good for magnetic materials. And as such, it's still one of the probes that is used extensively by hard condensed matter physicists studying magnetism, for example. Neutrons are very good at that. You could polarize them. You could do, like we said, inelastic scattering. And the very interesting thing, as far as this talk is concerned, is that they're differentially sensitive to isotopes of the same element, obviously. So, so you could see here that the scattering ability, like a cross-section, I'm just calling that as the scattering ability. Go ahead.
Yeah, so I just wanted to, what is the material of this kettle? Is it a metal?
Yeah, it's actually, I think it's probably, these were made out of aluminum, and aluminum is very transparent to neutrons. But neutrons are transparent, many materials are transparent to neutrons, and they're not as predictable as X-rays, which scatter as a function of number of electrons. Neutrons, before you actually had to calculate, actually do the experiments for everyone. Now you could actually calculate some of the cross sections, but before the cross sections had to actually be experimentally observed, unlike X-rays.
Because X-ray you cannot see through the metal.
Well, that's right, but aluminum in today's synchrotron sources you could do a better job, but steel you may not be able to do as. So they will be absorbed because they're interacting with electrons, while neutrons are interacting with the nuclei. So as you can see here, the scattering of hydrogen is very large for neutrons versus X-rays.
How are these images recorded?
This is just it's a shadow. It's not. It's a two-dimensional image. You can make. Also, at the end of the close of this talk, I'm going to run a little film that shows tomography, which is how you do a bunch of two-dimensional images and reconstruct them into three-dimensional image. So I'll show that. But that's just a film, like having a film, and it just shows you that because the water here has a huge amount of incoherent scattering, then it never makes it, let's say, to the film, to the detector, because it's scattered incoherent. It's not absorbed. You could argue, you can't really tell the difference in this case, but effectively it's scattered out of the detector. And this is usually made out of plastic or some form of polymer material, which is rich, again, in hydrogen. And because hydrogen has a very large incoherent scattering, it's a big noisemaker, then it scatters the beam out. And here is, I have to get an iPhone or something like that and show two things, because this is a camera. And nobody ever uses them now. You have to go to a museum to see them. But you can see how X-rays and neutrons are complementary, because the contrast that they offer is different. So here we have two different contrasts of the same object, and you could see that there's nothing here, but there's something here. And that is the film, and that is high in protium, in hydrogen. And of course, you could see many other features in the X-rays that you don't see in the neutrons, and so on and so forth. So they're very complementary. So when you're using X-rays, it would be good to use neutrons if you can, and certainly vice versa, because X-ray sources, of course, are much more plentiful, much more powerful, and so it's easier, they have easier access compared to neutrons. So neutrons are sort of a secondary technique, especially once you have something to look for specifically. It's a great probe. And we'll talk about some of what do you need to do it as far as soft materials, either polymers or biological materials, which fall in the same category. So one of the things that everybody knows, whether you do any kind of scattering, is that you require contrast. And then contrast you get in many different ways. So we could see each other because we have contrast. And if we sort of were monochromatic, then we would have a hard time differentiating us from the background. And so with neutrons, you can exploit this by using then different isotopes of a given element. And in the examples that I'm going to present to you today, we're going to be using protium, which is the most abundant isotope of hydrogen, and deuterium, which you actually have to make somehow. In most cases, you obtain it and then use it. It's much less abundant in nature. So here's just an optical description of how contrast is used. In this case, we're using something that has the refractive index the same. The beads have the same refractive index as this liquid. When you put them together, the beads disappear. In air, of course, the air has a different, and then you see them when they're outside. And this is basically what we're going to do in experiments. And this is one of the real powerful things that neutrons is, how do you change then the contrast of your material to understand then what you're interested. If you're looking at a given moiety, for example, you may want to be able to highlight it. So remember this, but this is wherever, here is something as a given material, DNA, for example, in D2O. And DNA will become invisible to neutrons in D2O at around 70% D2O. So if you had DNA in solution, it will scatter at every possible H2O, D2O condition, except when it's 70%. then it becomes effectively invisible, similar to these beads. And you can see wherever there's a protein, it will scatter, become invisible at 42%, lipids around 11%, etc. And the only reason that these lines are changing is because there are exchangeable hydrogens in water. And so as you're changing the H2O D2O ratio, those lines are not flat. If they were actually not exchangeable protons, then these lines would be absolutely flat. It's okay. So how do we make then neutrons? Aside from how Fermi did it in 1942, and then we have a guy to cut the rope and the cadmium falls in. So what we have are, at Oak Ridge, we have then the high flux isotope reactor, which is, as the name implies, it's a reactor source built in 1966, I think. And it produces neutrons by effectively the fission of uranium-235, which is the fissile atom nucleus of uranium, not 238. So 235 is what they use. And when people talk about enriched fuel, it means that there's a lot of 235. It depends on how much 235 you have in your fuel. So then you get effectively a neutron that will come and create fission products and produce, for every neutron, you produce about, on the average, about 3 new neutrons, which then go on to interact with other uranium nuclei and so on and so forth. And that is what is called a chain reaction. And this you want to control, because if it's uncontrollable, then you have a bomb on your hands. So the reactor is physically right over here. There's a lot of other facilities. And the other facility I'm going to describe to you is over here. This is called the spallation neutron source, and that's where I live most of the time. But over here is the reactor. The core, as you see, is very small compared to the infrastructure, the rest of the infrastructure. And here we have then all of these instruments away from the reactor, and including stuff around the reactor. These are called thermal instruments because effectively what you're getting are neutrons that have been thermalized, and they have wavelengths of around a few angstroms, 2, 3 angstroms or so. And you could manipulate that because you get sort of quite a few neutrons, a spectrum, and then you use monochromators or the like to then choose the wave, the window of wavelengths that you would like. But if you actually cool them down in a bucket of liquid hydrogen or deuterium, then you slow them to the point that their wavelengths now become much, much longer, which is suitable, more suitable to studying materials like biological materials that have large structures versus hard materials that normally have very small structures, repeat units. And then you could transport these neutrons using various optics to quite great distances, and then you use these other instruments over here, which are far away from the reactor, and these are called the cold instruments, and that is called the guide hall. And the security in here versus in here is very, very different. So the cold instruments, you could sort of walk in and out. The thermal instruments, it's a bigger deal to go in and out. So that's one way of producing neutrons. And these instruments, the blue, are all of these type of production, reactor production. And at the Baba, you guys in India have a reactor sources. I don't know how many and what the neutron situation is, but I know that there are neutron scattering facilities there. So the other thing is the spallation neutron source, which was in the distance in the other picture. And this is effectively an accelerator-based source and uses a liquid mercury target. And here what you have is a proton that is 1 GeV that interacts then with mercury, which has a lot of neutrons. It breaks it up, through a process called evaporation. And effectively what you get is about 20 neutrons per interaction. The problem is that these are pulsed, so it's not a continuous situation as you're getting in a reactor, but it comes with time stamps. And of course, you could use these time stamps to your advantage if the experiment requires it. Now, spallation source neutrons, if you integrate the flux versus reactor-based neutrons, you always get more neutrons in a reactor versus a spallation source. The thing is that your pulses here are much more intense than the steady stream of neutrons that you get from a reactor. So if you want certain events and you could pulse it just right, you could get very, very good signal to noise. And you have now some kind of timestamps associated with your experiment.
Is there any way to pollinate the neutrons?
Absolutely. So people collimate them. You could change their wavelength. You could choose whatever chunk of wavelength. With the spallation neutron source, you could do it better because you have timestamps. With the reactor sources, you have to do more mechanically. So here you could collect all the data and then choose what you want with a spallation neutron source. Because, like I said, you have effectively a every neutron has a timing event with it. It has a stamp. And this is where we're going now with the SNS having a flux up here, and J-Park, and in Europe they're having the ESS is coming online in the next few years. But they've been saying this now for quite a few years. So we'll see how that goes. So what I want to talk about is membranes, and I know there's a lot of work on membranes here in Bangalore. Various groups are working on it. And we had a meeting, a workshop, a 2.5-day workshop on membranes. Ganapathy had that. We went from Thursday to Saturday, and that was great. And this is the stuff that I've worked on my entire career. I've done X-ray scattering. I've done scattering effectively, neutrons and X-rays, but I also use a lot of other techniques as I'm doing now. But lipids have always been my mainstay as far as science is concerned. And what is a lipid? It's effectively a fat that has a hydrophilic head group and hydrophobic chains. And the interesting thing is it took us a long time to understand how a membrane is organized. So the first time we actually learned, even though we knew about membranes, the first time we knew that there was a bilayer associated with this membrane. Because membranes go back to the 1700s, people were aware of them. But the fact that we had a bilayer is in 1925 by an experiment done by Gorder and Grendel, who basically took red blood cells, calculated the surface area of the red blood cells, then laid them on a film, and then calculated the area of the film, knowing the area of the red blood cells. They ended up coming out that this has to be a bilayer. These lipids have to be a bilayer. And that's because the red blood cell only has one membrane, so you know that all of them have to be associated with a plasma membrane. Now, they made many mistakes, but they seemed to cancel each other out, and they got the right answer. So, such is life. Then we go to 1935, where the proteins come into it, and because of electron microscopy was just developed around that time in the 30s. And people were seeing these very interesting lines. And of course, they knew about the existence of proteins. And so they thought, oh, well, the proteins must sandwich the bilayer. So that was the next iteration. And then we go to 1972, which is closer to what we think is happening today by Singer and Nicholson, because there were so many other studies that were contradicting this 1935 structure. So we come to this called the fluid mosaic model, which effectively says that the membrane is a two-dimensional liquid with proteins embedded in it and freely, and they're allowed to diffuse freely in the two-dimensional plane. And then a few years later, so these are a few years later, it's quite a long time, right? We started in 1925, and now we're in 1997. And what we're saying is that, no, these things are not they're not just two-dimensional homogeneous fluids, but they're actually mosaics with these so-called lipid domains that then enable functionality, to impart functionality to the proteins. So this was a very big question for many years, and I think it is today, but people are just tired of looking at nanodomains in some ways. And when I went to Oak Ridge, what do neutrons do? So we had to find a problem that was relevant for neutrons that X-rays could not do, because X-rays, as we said, are much more powerful than neutrons. So if you could do something with X-rays, of course you would do it with X-rays. Why would you do it with neutrons that have, 10 to the 7, 10 to the 8 less neutrons compared to X-rays? So, as I showed you before, the real nice thing about neutrons is you are able to manipulate the contrast much better than X-rays. And until synchrotrons came around, it was very hard to manipulate the contrast with X-rays. And even now, you still need something that you could do anomalous scattering, which means something quite heavy, or reasonably heavy, to get then contrast with X-rays. I mean, beyond the initial contrast. In other words, manipulating it beyond the ground, let's call it the ground state experiment. But you need then isotopes of that element. And in our case, because these things are loaded with hydrogen, obviously hydrogen is a good element to work with. And also you would like to get rid of protium, because protium is a huge incoherent scatterer, which means it adds to the background, it adds to the noise. And you'd like to get rid of it if you can. So here we are. We're going to think about this as the neutron experiments that I want to describe to you as fluorescent experiments. So here are domains floating around. This is, of course, an artist's conception of what domains look like. But assuming that that's it, you see that there's a thickness mismatch between the domain and the rest. So that's a thing to look for. And as you can see here, this is simply a bunch of lipid molecules making a giant metal vesicle, and only certain of those lipids have a fluorescence label, right? So you see they're all sort of interdispersed, and then as they're phase separating, because you lower the temperature, then they start to form then these domains. And of course, it's easy to see them under a microscope when they're microscopic. But what happens then when they're nanoscopic? Because the real controversy is that nobody had seen lipid domains in biological systems. So it was coming to the point that either they don't exist and people are making it up, or they're very, very small, or they're ephemeral. In other words, they just simply come and go very, very quickly. So what is it? So we're going to do effectively this experiment, but with neutrons, and looking at nanoscopic scales instead of the micron scale. So the only thing that you have to know about this machine here is that neutrons come in. I don't know if actually, oops, I'm very bad at this sometimes. I'm sorry. I'm not good at pausing. So as you can see, there are neutrons. This is a velocity selector. It comes in, then you could collimate it. And so the beam size is changing. Then it's collimated again, interacts with the sample, and then it goes into the detector. So it's not unlike a light scattering experiment in many ways, except in a light scattering experiment, you have a laser, which is already telling you what the wavelength is. Here you actually have to manipulate the wavelength using effective many ways, but one of them is called a velocity selector, which has fins that are curved. So it will only allow the neutrons with the right velocity to go through when it's spinning at a given rate. So if you change the spinning rate, then they will actually collide, because the curve on those for a given wavelength, the curve is only made for that wavelength. So then you will get different wavelength neutrons coming through. So do you follow it? Because it'll do this. So the only thing you have to remember is that scattering is dependent on a few instrument terms. So your detector efficiency, your solid angle, etc. Number of neutrons, for example, does count, as we said. And then it depends on this differential cross-section, which is given by then your sample. And that is something that is very important. So that is your sample terms. And one of the things that you see in here, of course, is the contrast of your sample. So that is effectively neutron scattering. So if we don't have any contrast, then we will get zero scattering. And if we have contrast, then we will see something. Right? That's all it means. So so what we decided to do, I had a postdoc when I went to Oak Ridge in 2010. He started in 2011, so we both effectively started together. And I had worked on lipid domains when I was at Chalk River. And he also had done it, but with fluorescence. And he had a lot more experience with different systems than I did. And so we decided to look at some of these systems in some clever way that we could see, can neutrons detect then nanoscopic domains? And what does it all mean? So what you do is you find a mixture that has been studied quite well, a canonical mixture that will phase separate. And in this case, we settled on DOPC, which is biologically irrelevant. It's not found in biology. It has 2 unsaturated fatty acid chains. POPC, which is actually very common. Every time I eat butter or oil, you got a lot of POPC. And cholesterol, and then we chose this lipid that will phase separate into a particular phase called DSPC, and that is deuterated. So that is a deuterated lipid that we use to give us contrast when it goes into a particular domain. So remember the light scattering, the fluorescent ones all went into a domain, and the other ones you didn't see. It looked like there was no lipids and these domains were floating, but no, they were floating on the surface of a giant vesicle. So we did this experiment by effectively taking this mixture, starting with POPC cholesterol and chain per deuterated, and then slowly titrating, taking out the POPC, taking out this, and putting more of this, to the point that then you had 100% of this DOPC versus POPC. So we did that, and as you can see, when you have POPC, pure POPC, cholesterol, and DSPC, those 3 lipids, you could see that there is a small amount of scattering. So the way he did the experiment is it was very clever. What you wanted to do is take these vesicles at every different concentration, and put them at a high temperature, whereby all of these things mix uniformly, right? So you have them mix uniformly, and then you contrast match them, meaning I put now the right amount of H2O, remember those curves? That we put the right amount of H2O, D2O to make that entire vesicle invisible to neutrons. And this is what he's showing over here. This is basically background. And then what you do is you lower the temperature. That means they will phase separate into domains, and then you record that. So you basically kill all the scattering at high temperature, and then hope if there's phase separation, you will get then a signal. If there's no phase separation, you will continue to get no signal. So what he did is he is now titrating, changing the amount of POPC for DOPC, and you can see that there's more and more and more and more scattering, which means that it's forming bigger and bigger and bigger domains. Now the thing about neutron scattering, unlike microscopies, it's not a real space technique. Unlike X-ray scattering, they're not real space techniques. They're reciprocal space techniques, which means that in order to, unless you're doing holography, neutron holography or X-ray holography, there's no way that you could go easily from reciprocal space into real space. So in order to go from reciprocal space to real space, you have to create a model and then fit the data to this model. And that then is hopefully the correct answer based on other things. So he does this using Monte Carlo simulations, and he finds out that the domains in the biologically relevant POPC are quite small, and they increase in size as you're going into the biologically irrelevant phase by quite a bit, which means that what he saw is that the LO phase, the liquid ordered phase, is not changing in thickness, but the liquid disordered phase is becoming thinner and thinner as a function of this the other lipid, which means that the mismatch between the domain and the surround, now there's a thickness mismatch, which means now you have a line tension in there that drives then the formation of these domains. That's what we think. And this was the first experiment to sort of show how line tension, because theoretically line tension has always been there. It's one of the ways to create and stabilize membranes, but this was sort of the first experiment on nanoscopic domains to show this, whereby you're now changing effectively that line tension by, it's increasing, and the thickness mismatch between the two phases is also increasing. So we know that we could actually see nanoscopic domains, but we actually had to use, deuteration in order to do that. Because if we didn't do it, then everything looks the same, right? So if you look in biology, you could say, okay, how do we do a biological system that we could do the same sort of strategy? Well, in the 70s, the easiest way to do that was to look at certain types of viruses, because some types of viruses only have two components for the most part. They have a portion of RNA and a part that is protein, and that's it. So in theory, those are fairly easy instruments, samples to look at if you want to study, for example, the RNA part or the protein part separately. You could also look at it both at the same time. So if we wanted to look at the same time at the whole thing, I could go anywhere where there's no RNA or where there's no protein. So if I avoid 60 some odd percent D2O, and I avoid 40 some odd percent, every place else, every other concentration will give me the whole structure. Now, you would prefer to be closer to 100% because you will get less background from D2O than H2O. So people want to work closer to, because most of your sample will be made out of water. So you want to work closer to the D2O rather than the H2O, because the background will be much, much greater at 100% H2O. So what they did is they effectively contrast matched, did experiments at 62 and at 42 or something like that, whatever that is, 70. And you're able now to look at the RNA separately from the protein. So that's interesting. You only have two components. You don't even have to deuterate. You don't have to do anything. You just have to change the H2O-D2O ratio and basically hone in to what you want. And what they did is exactly that. So these are the scattering patterns from 69.5% D2O, and that will kill effectively all of the RNA. You'll be looking at the protein shell. And then you do at 42, and then you're looking at the RNA core. And then if you take effectively a Fourier transform of the intensity into a radial distribution function, you see that 100%, where there's no contrast matching, what you see is a lot of peaks. Right? So there's a lot, it means that the correlation, there's some correlation at this distance, there's something there at that distance, there's something there at this distance, and something there. But if you actually then do at 42%, you will see this is how the distribution of the protein. And at 69.5%, this is the distribution of the RNA. Right? I'm sorry, This is the RNA, and this is the protein. Because don't forget, 42%, this is where you contrast match out the protein. So that's a simple system, right? And you could do that. But what happens now if somebody said to you to use a whole entire cell, which has RNA, protein, lipids, cholesterol, this, that, like they say, the whole catastrophe is in there. What do you do? Well, that's a much more difficult experiment, and that's why you see a lot of model studies with scattering, and you don't see real systems. So we decided around 2016, 2015, maybe 2015 or so, to see if we could actually look at a system like that, just to see if we could see, tackle. And one of the things that I ended up interacting is with a bunch of biologists who actually know biology unlike me. And I said, okay, would it be possible to look at nanoscopic domains in a real system, a fully functional system? And they said, of course, what are we going to look? Because you have prokaryotic cells and eukaryotic cells. And the thing is that with prokaryotic cells, bacteria and the like, you're able to manipulate them quite easily because they're survivors. Eukaryotic cells are very sensitive. And one of the things that you could do with prokaryotic cells is you could introduce a lot of deuterium in them, and they will still be functional. They may not be the happiest. They may actually change their ways, but they'll still be able to function and replicate. If you do that with a lot of eukaryotic cells, then you got yourself a problem. And that's why it's not recommended that you drink, besides being expensive, heavy water. Now, don't drink heavy water, because if you drink heavy water, it's not going to be good for you. And that's because we're very sensitive to the dynamics and et cetera of water, the hydrogen bonds, and does all kinds of things. So these small changes in D2O and H2O affect eukaryotic cells to a great extent, and much less so than prokaryotic cells. So they said, we need to look at prokaryotic cells, all right? And then when you're dealing with some of these, like these bacteria, you have gram-positive and gram-negative. Well, the easiest thing is to work with the gram-positive because you have one less membrane. So you want to minimize the complexity as much as you can, all right, instead of having two membranes over here. And of course, you have this peptidoglycan layers on the outside. So we decided to look at this bacterium called Bacillus subtilis. And Bacillus subtilis has been studied to a very, very large degree by biologists. So they know lots and lots and lots and lots of things about it. So we went into this, and if you take now living bacteria and you put them into the beam, you will get a scattering curve that looks like this. In other words, you will get intensity. So you're integrating the whole signal on the detector, just straight ahead, integrating the whole thing. You will get a lot of intensity at 100% H2O. And as you're coming down to D2O, 42%, it's a dip, but there's still stuff. There's still intensity. It's not killed. And then it goes up again. Why is that? Why is that? Why is there a dip? Well, the bacterium is mostly made out of protein. So it's got a lot of protein in it. So that's why you see a dip, because you've contrast matched out the protein part. Right? So the protein now of that bacterium is invisible. But of course, there's a whole bunch of other molecules. That's why it never goes down to zero. Right? In the previous experiment we did with the vesicles, we were able to go down to almost zero, because we only had lipids, and if they mixed them all correctly, and you put them at that contrast, you could kill all of it. But you can't do that here, because it's got RNA, DNA, and all of the other stuff that we talked about, sugars, cholesterol. In this, not cholesterol, here they have different types of sterols. So what you have to do is then do some tricks to them. So this is the trick that Jim Elkins and Jonathan Nichols did, is you take these bacteria and you could grow them in 100% deuterated media. In other words, they feed off of whatever they take in and synthesize. It's going to be deuterated. So you give it, I don't know, sugars or whatever they gave them. I can't remember. And then what you want to do is somehow then introduce then fatty acids that are proteated into it. But that's not simple, because bacteria, you could grow them, and they will become almost 90% or so D2O. And then if I fed them proteated fatty acids, they would take them. But then they would do all kinds of shenanigans and change them anyhow they wanted to, and cut them up, and then you won't know what you've got. So they would do all kinds of interesting tricks to them, including changing their state of deuteration. So all of this stuff then means that you got to do something else. So what you do is you use, effectively it's a poison, it's called cerulenin, but you could do it with CRISPR today, where you look at the factory that is making fatty acids and degrading fatty acids, and you shut those factories down. Those mechanisms are shut down so that then it becomes, go ahead.
Did you use transformation in the past?
Hmm?
Did you use transformation in the past?
What does that mean?
Transformation. Instead of CRISPR, you can use gene editing.
Well, you could, I don't know. I mean, CRISPR is one of the things that people have been trying to use, but you basically use cerulenin to actually knock out those pathways. Now, you could knock them in different ways. That's fine. But the moral of the story here is you got to knock down those pathways so that they cannot make fatty acids or degrade the fatty acids, those two things. So that's what you got to do. We did it with cerulenin. The problem with cerulenin is it's tricky to work with, and it's very expensive. And maybe the technique that you're proposing to me would work quite well, or CRISPR. You got to do something to effectively shut those pathways, because then you're able to control what you could feed to them. That's the whole idea. So it's not because we want to be mean, but in order to feed them, and they will then take those fatty acids that you need to shut them down. Otherwise, they'll do whatever they'll do what they want. So once you've shut them down, then they're dependent entirely on you to feed them the fatty acids that they require. And of course, we know that they only have one membrane. And the interesting thing is that we only had one fatty acid that we could deuterate. And this has 7 fatty acids in total. So they have 7 fatty acids, and they could make any type of lipid from those 7 fatty acids. In our case, because we only had one fatty acid that we had deuterated, I can't remember if it was, I think it was probably palmitic acid instead of anti-iso 15-0. And they were the 2 essential fatty acids, meaning they require those fatty acids to survive. You could take the other ones away, but they require those 2. And it's interesting. Now, instead of using 7 different fatty acids to make a lipid, the lipids that they wanted, they're using 2 fatty acids to make all the lipids that they wanted. So the lipidome has probably changed, because now they have to maintain some homeostasis for the whole system to work. So when you do that, and then you feed them the fatty acids, the proteated fatty acids now you've created what? Contrast, right? Because the whole thing is sitting here. So you can see now where the whole bacterium is sitting. You've shifted all of those proteins and RNAs and all that from the other diagram that we had all the way to a single point almost, where you could now entirely make the entire bacterium invisible to neutrons. And then what you do is you reintroduce fatty acids, but now they're proteated, so now they have a very different scattering length density. And what you want to do is shut down, make everything look like this, except for the membrane. And this is exactly what we did. We did this particular experiment, and then you do a scattering, and this looks exactly like if you had some planar membranes, and you're able to fit them to effectively a 2-dimensional membrane in a living bacteria. The interesting thing is And then we actually went on to,
So you're only changing the contrast, you're not deuterating any of the lipids in the bilayer?
that the whole bacterium is deuterated because you've grown it under deuterated conditions, so it has become now deuterated. Its lipids are also deuterated, but because now we've shut down its mechanism of creating new fatty acids and also changing the existing fatty acids to something else, otherwise they will do whatever they want. You feed them then proteated fatty acids. They will take them and make whatever lipid they want, but the lipid will always have fatty acids that are proteated because that's the only source that they have to make lipids from.
That's what gives you the contrast.
And that's what gives you the contrast. Because otherwise you would have. So now we have a whole system that is deuterated except for the fatty acids in the bilayer. And then you're able to fit it as if it was a model system. A Q to the -2 dependence in the mid-Q, and at the high Q, a Q to the -4, which effectively is exactly what you would get if you had vesicles, traditional vesicles. And then we did some more tricks to actually find that they actually do have 40 nanometer diameter domains, and that was more effectively contrast studies that we did, and I don't want to get into it. So this was an interesting thing that you can actually now have a platform to look at nanoscopic things in a real system. Now, of course, you can't apply it to too many systems. As we said, with the eukaryotic cells, you couldn't do it because you can't bring them to that level of deuteration. So the other thing is that, people did not believe that the literature effectively did not have any papers that showed nanoscopic domains in prokaryotic systems. They only thought they happened in eukaryotic systems. And we, along with others from Harvard, showed that, yes, the nanodomains exist in all kinds of systems. And I think that makes sense, because if they're that important, why would they only exist in one and not the other? There's a lot of similarities. So that was an interesting thing. And effectively, it takes a lot of work to do it. But if you have questions on the nanoscale, then this is a great platform. And there are many techniques that now, besides what we did, CRISPR and gene editing, other techniques that you could use to shut down pathways more effectively than what we did. So that was it, and I'm going to finish off with a tomography experiment. So this is, you'll see. And I will be happy to entertain any question or about the work that Raghu was talking about, neuromorphic materials that we're working on now. That we haven't done any neutrons, but we will. So thank you very much, and let me know if you have any questions. So look at that. So that is a 3D printed turbine blade. And then effectively you take slices and then reconstruct them in the computer, but you don't have to destructively break up the material, right? It's just a tomograph of that material.
What is the dimensions of this?
I think the dimension, I think it's something like that. Yeah, it's like a few centimeters, 5, 6 centimeters. So these are, they have all these cooling channels. So I think that's one of the things that they're worried about, getting into those cooling channels.
So what would the scale bar be over there?
I think the resolution of the neutrons is on the order of microns.
Okay.
Yeah, microns is the resolution. Yeah. And of course you could do that with people do it with X-ray tomography and other in very, very nice ways. In fact, the resolution that you could get from X-ray tomography is much, much better, much better. I mean, it's amazing some of the things that they do.
Okay, because slices by putting detectors at
Yes, yeah, and orienting the sample, yeah. So yeah, you have to take it from different angles, yeah. So one of the things you could do is just keep the detectors and then spin the sample. Yeah, so that's the advantage of neutrons is really you have to be able to manipulate contrast, and you could do that much easier than with X-rays. Now, of course, with X-rays and synchrotrons, because you have a tunable beam, you could do it quite well, but you need something heavy with a lot of electrons in order to get anomalous scattering. With light materials, you can't. You know, you can't do it with hydrogen. There are no synchrotron sources that could do that.
We'd be happy if you told us something about neutron therapy for cancer or tumors.
I mean, I don't know very much about that. I mean, boron activation is the thing that they've been using. So I think what they do is they impregnate some boron particles and then they hit you with thermal neutrons, and it's. But I don't know where it's at. It's not something I know much about, but it's something that's been ongoing for, I don't know, 40 years, 50 years. Unless some big breakthrough happens, which there may have been, I don't know. You know, it's a thing that just kind of goes along. Now, you could argue that cryo-EM was like that until, you know, the new detectors came along and the new algorithms for orienting the particles. So people think cryo-EM is something new. Well, it isn't new. In fact, electron microscopy goes back almost 100 years. Cryo-EM goes to 80, in the 80s, somewhere there. They started to protect samples. And really, it didn't go anywhere until 10 years ago, I guess, when all of the technology. And so things can happen sometimes unexpectedly, but I don't know what has happened with neutron therapies. And again, the other problem with neutron therapies is, what do you know, it'd be a big deal. There are not many neutron sources unless you have some kind of portable source. And you don't get a lot of neutrons from portable sources as far as I know. So it's a problem. One way or another, it's a problem. Go ahead.
Hello. Yeah. So if rather than using deuterium, if you were trying to change the carbon itself with a heavier isotope, would that affect the experiment significantly?
Well, you have to look. See, the thing about hydrogen is it's ubiquitous in soft materials, and biology is a soft material, and there are lots of it. So it makes sense to change it. And the reason, if you keep it and let's say you change something else, it's a huge incoherent scatter. In other words, it creates a lot of background. It makes a lot of noise. It's a big noise. It's the biggest single incoherent scatterer of any other, compared to any other nucleus. So that's why you also work in high H2O, D2O conditions, because in many cases your sample is mostly water. So if you put H2O, it's going to create a big, big problem for you to extract the noise, you know, the signal from, because it'll be buried under the background. So, you know, there are many ways to do things, but in this case, H2O, D2O is a big deal, and that's the way to deal with a lot of problems. The problem is then making samples. That's another question altogether, and we're proposing to create a national facility to make samples so that we could enable the neutrons to their best of their abilities. Because if you don't have this contrast, they're no different than X-rays. Right? There's just no difference. You wouldn't see any of the things that we said. You would see some average of lots and lots of things. I mean, the virus is, that particular class of virus is just an exception, you know, where you only have two components for the most part. Okay, well, those, you just have to change the H2O ratio and you could see both. But that's an exception. You know, there's always an exception. The thing is, what is common and what can you do for for your everyday user? You need deuterated or isotopically labeled, you know. And even in X-rays, you still sometimes, when I used to do X-rays in another life, and I wanted to locate things, I would put bromine in. So you would look at bromine, where is the location of bromine, and you used it as a proxy for that molecule. Because bromine got a lot of electrons. So it gives you contrast.
Hi. So in the same sample, in the same lipid bilayer, if you have lipid domains with inhomogeneous size,
Yes.
can we characterize them quantitatively?
Yes, yeah, because basically you will get scattering from if they face separate, even if they're different size, you'll get scattering. Now, in order to analyze the size, you'll have to fit the data in various ways, because none of these things is 100% for the truth, right? That's what it fits the data. So you have to minimize, have a complementary data, then that could, minimize then the type of models that you could use. So yes, you would see them, even if they're different. Now, your signal, interpreting the signal may be more difficult.
So thanks for that very nice talk. You told us mainly about structure, and of these very, very complicated and heterogeneous systems. My question was about dynamics, say
Yes.
using neutron spin echo.
Oh, yeah.
So what is the status of that?
Okay, so I have some data here. I didn't show it to you. Let's see if we can get to it. So again, so this is Fred. This is Fred Heber. Whoops. Let's see. So this is Fred. So he's the guy that did the nanoscopic domain in vesicles, and Jonathan Nichols. So this is yes, neutron spin echo. So neutron spin echo is another technique to do dynamics, and it's becoming more popular, partly because the dynamics that it's looking at falls in the regime of molecular dynamic simulations. So they're very complementary. You're looking at motions from 10 to the 9th and something like that. And length scales also, you can measure some length scales. But effectively, it looks at the slower dynamics of it's backscattering or something like that. So it's an instrument effectively that you have 2 NMR magnets, right? You have 2 NMR magnets flip on their side. And then because, as we said, you could polarize neutrons spin up and spin down, you could then choose a particular spin of neutrons, which means that you're throwing away half of the other neutrons, right? So it's already, the signal is weak, now you're making it weaker because now you're choosing a slice. And then you put it into a coil that precesses it, zips the neutron up, and then it interacts with a sample, and then the next NMR machine unzips the neutrons, and if the polarization has not changed, meaning that you started with up and you end up with up, that means nothing has changed. The whole thing was elastic, there are no dynamics going on. If it now has changed in polarization, then something has gone on, right? And that's how you measure then the dynamics, and they're looking at collective motions on the order, like I said, 10 to the 9 seconds or something like that, 10 to the minus 9. So this is something that you could use then to look at the breathing modes of materials. And you could get, one of the things that you could get from them is the bending rigidities. So what you do is then you do a bunch of experiments whereby you could collect at a given angle then the various data sets. And as you could see, if there's no dynamics, they would just be straight lines. And because they're falling off, that means there's something going on, and then they're falling off there's differences between angles, so that's giving you a length scale of that these things are happening at. And then you use then some kind of model to fit the data. Now, all of this is, you have to believe that the model is correct, and there's a lot of debate these days with regards to, is the model correct or not, or do we need to make certain assumptions to fix the model? So yes, there are many neutron and elastic neutron scattering techniques, and I think for the most part, neutrons do a better job than X-rays on that, although I stand to be corrected these days. But yeah, so neutron spin echo is one of those techniques that is sort of gaining popularity because it falls in the right time regimes and length scales that you could look at materials. So, it's another question. It's another technique, and if you have that particular question to answer, what are my bending moduli? And I don't have any other data. Yeah, we did. So we looked at all of these. So Jonathan Nichols looked at all of these things. The domains, the bending rigidity of domains. And again, you had to use neutron contrast, the whole vesicle, the surround. So he did all of these things, and then he combined it with Shaolin Chen with then simulations, where you get then, I don't have that, but where you actually get then a molecular, using this data, you actually then get a molecular picture of what's going on in the bilayer. And especially you see interesting things happening at the interfaces. So yes, there is. There's a really good machine at ILL, and we have a reasonably good machine, and I think they're talking about building another one at some point. But yeah, neutron spin echo is one of those techniques.
Are there any other questions? If not, let's thank John once again for a very
Well,
interesting.
thanks a lot.
They used a lot of this for water, right?
Huh?
For water, neutron spin echo for water dynamics.
I don't know. They
When
don't
you have
use
water in your
it.
surfaces and things, no?
But they use it for effectively the collective motions.
Yes. How much of this?
Thank you very much. I appreciate your help. Thank you. My gizmo, before I forget, I've made a note. Oh, there it is.